Protein folding crisis hotline. You folded, we fix it. What's your emergency?
Okay, hi, hello. So I think I'm folding wrong because I was supposed to be a functional enzyme, but suddenly I'm clumping up with others, and suddenly I don't look right. I don't look like my parents, and then am I turning into an amyloid? Oh my gosh.
Okay, stay calm. Deep breaths. Are you forming beta-pleated sheets?
Yes, but they're everywhere.
Mmm. Okay, hold on. Sounds like you got a classic case of a misfolding, man. Don't worry. We're about to unwrap exactly what beta-pleated sheets are, why they matter, and how to spot the stable ones from the troublemakers.
Okay, then will this at least help me not turn into a prion?
Well, that's up to you. Only if you pay attention.
Huh. Okay, how about how
Hello everyone, this is Group 9's lecture video, and we will be talking about one of the secondary structures of our protein, specifically the beta-pleated sheets protein structure. And further into this video, we will be talking in details, especially our other group members, the different aspects of the topic itself. So here are our learning outcomes. So by the end of this lecture, hopefully you'll be able to define what beta-pleated sheets are, explain how hydrogen bonding stabilizes the beta sheet structure, differentiate between parallel and antiparallel beta sheets, recognize different variants, your twisted beta barrels, and etc. Relate beta-pleated sheets to disease mechanisms and clinical applications. And hopefully by the end of this video, our client earlier will be able to solve their problem. So now I put the stage to Mr. RONDRIQUR.
Good day, I'm RONDRIQUR, and today we're watching Protein Unwrapped. With me today are three incredible educators, each unraveling a part of this mysterious structure. First up, let's take it to our field reporter who's gone deep into the hydrogen bond trenches. What does silk, prion diseases, and your future biochem exam all have in common? The answer is beta-plated sheets. What is a beta-plated sheets? A beta-plated sheet is one of the two major types of secondary structures found in the proteins, the other being the alpha helix. The beta sheets is characterized by a sheet-like arrangement of polypeptide chains that lie adjacent to each other. These strands can run in the same directions or in the opposite direction. The name pleated comes from the zigzag or accordion-like folding pattern that results from the specific angles between peptide bonds, the stabilizing forces of a sheet. So first we have hydrogen bonds. The primary stabilizing interactions in the beta sheets form between the carbonyl oxygen of one strand and the amide hydrogen of another. These bonds are interstrand, meaning they occur between adjacent strands and hold the sheets together. Antiparallel beta sheets typically have more linear and stronger hydrogen bonds than parallel sheets. Second, we have the van der Waals interactions. Weak, non-covalent attractions between closely packed atoms. Since the beta sheets have tightly packed side chains, these interactions contribute to their overall stability by allowing the strand to fit snugly together, especially important in the core of globular proteins where space is limited. Lastly, we have hydrophobic interactions. Non-polar amino acid side chains in the beta sheets often orient themselves inward in a protein away from water. When multiple B sheets are buried inside a protein, hydrophobic side chains cluster together, excluding water and stabilizing the protein's folding shape. This is particularly significant in water-soluble proteins, where hydrophobic residues prefer to avoid the aqueous environment. So next is structures and stabilizing forces of a beta sheet. The beta-pleated sheet is composed of two or more beta strands, which are segments of a polypeptide chains that are stretched out rather than coiled. So these strands are positioned side by side, forming a sheet-like array. Each beta strand is typically 5 to 10 amino acids long and lies in a nearly fully extended conformation. The side chains of the amino acids in each strand projects alternately above and below the plane of the sheets, allowing for tight packing and interaction with the surrounding environment or other protein domains. The orientation of the strands determines whether the sheet is parallel. A strands run in the same N to C terminal direction, resulting in the angled hydrogen bond. But where there is structure, there is controversy. Let's hand it over to Saavedra.
For the structure and stabilizing forces of a beta sheet, the sheet consists of beta strands connected laterally by hydrogen bonds. Each strand is extended rather than coiled, allowing side chains or R groups to alternate above and below the plane of the sheet. Sheets may be parallel or anti-parallel depending on the direction of peptide chains. For the stabilizing forces, hydrogen bonds, van der Waals interactions, and hydrophobic interactions. For the first one, they form between the carbonyl oxygen of one strand and the amide hydrogen of an adjacent strand. For the second one, between tightly packed side chains in the sheet. And for the third one, or your hydrophobic interactions, they are in globular proteins. Hydrophobic residues on beta sheets may pack together away from water, adding stability.
And there you have it. That's how you define your beta-pleated sheets and also your stabilizing forces that comes along with it. Since that has been tackled, let's move on to how it differentiates with other structures, especially with the other secondary structure, which is your alpha helix. And then let's also talk about the other variants that is involved in your beta-pleated sheets.
Now, let's differentiate from other structures. Beta sheets, as I mentioned, are flat pleated structures formed by hydrogen bonds between strands. They provide stability, form the protein core, and support molecular binding found in strong proteins like silk and in disease-related aggregates. For alpha helices, they are spiral shaped and stabilized by bonds within the same strand. They are flexible and common in membrane proteins, DNA binding proteins, and structural frameworks. For random coils, they are irregular, unstructured regions that allow flexibility, common in intrinsically disordered proteins that adapt to different targets. For turns or loops, they are short segments connecting helices and sheets. They help protein fold compactly and are often part of active sites and binding regions. For the characteristics, roles, and functions, beta sheets are one of the fundamental secondary structures in proteins, formed when beta strands are linked side by hydrogen bonds. They contribute to protein stability and have diverse functional roles. So, they are made of beta strands aligned next to each other, can be parallel or anti-parallel, or stabilized by hydrogen bonds between backbone atoms, and have a pleated or zigzag appearance due to bond angles. Strong, silent, and twisted. That's how beta sheets survive. To know more about structures and kinds, let us now move on to Jewel.
Now we're gonna talk about the different variants of our beta-pleated sheet structure. So beta-pleated sheets are common and crucial element of protein secondary structure. It is characterized by extended polypeptide chains arranged side by side, held together by hydrogen bonds between backbone atoms as discussed by the previous presenter. These sheets are not uniform. They exhibit structural variations depending on the directionality and interaction of the strands. So the first two structures that we're going to talk about is your parallel and your anti-parallel beta sheets. By their name itself, it's actually quite explanatory. Your parallel beta-pleated sheets means that all strands run in the same N to C direction, basically from your amino to your carboxyl terminus. So it features hydrogen bonds that are angled and not directly linear. It is typically less stable than antiparallel sheets due to the less optimal hydrogen bonding geometry. So moving to your antiparallel beta-pleated sheets, The definition for this one is that the adjacent strands run in opposite directions. So basically they are facing, but they run in opposite directions, as you can see. So it features hydrogen bonds that are more linear in comparison to your parallel beta sheets, and this makes them stronger and more stable. So it is more commonly observed in proteins than parallel sheets. And the strands are often linked by tight turns or loops called beta turns. And then the example for this one is your crystalline protein regions, and they are your silk, your fibroin. Now moving on to our mixed beta sheets. So basically by the name itself, both of your parallel and anti-parallel strand orientations are here. So this occurs when a protein folds have its constraints. Thus, it creates a hybrid alignment. So basically what happens here, the hydrogen bonding varies across the sheets, making it actually moderately stable. And this is seen in complex globular proteins with irregular folds. An example for this one is very much common in enzyme active sites and binding proteins, where flexibility is key to its processes. So another variant are your twisted beta sheets. So these are not flat but exhibit a right-handed twist. So what this is the twisting arises due to amino acid side chain interactions and backbone strain. This conformation is more common than perfectly flat sheets in real proteins. This provides your compactness and flexibility to protein structure. An example for this one is found in many enzymes, such as your triose phosphate isomerase. So here is a tabulated summary with all of the variants, its strand directions, bonding stability, and where they are found. So while your beta sheets are typically shown as flat, zigzagging strands in textbooks, real proteins reveal a rich diversity of sheet architectures. So the directionality, twisting, and connectivity of strands affect the stability, its flexibility, and functionality of the proteins that they compose. So understanding these variants is essential in fields like structural biology, protein engineering, and drug design, which will be discussed further by our other member. So since that is covered, I will now be putting the stage to Salomon.
Function follows form, and these sheets fold to hold. The Beta-Pleated sheet is one of the most remarkable elements of protein architecture, strong, stable, and efficient. But what happens when this element structure goes wrong? When folding falters, the consequences can be catastrophic. Let's first explore why these sheets matter so much, and then uncover what happens when they misfold. The importance of Beta-Pleated sheets, first, is the structural stability. Beta-Pleated sheets plays a critical role in maintaining the structural integrity of proteins. Their flat, extended arrangement and their interstrand hydrogen bonds create a highly stable framework. This gives protein rigidity and resistance to mechanical stress, especially in tissues and biological materials that require durability. Second is the mechanical strand. A classic example of this strand can be found in silk. The protein fibroin, found in both spider silk and silkworm silk, is composed predominantly of beta-pleated sheets. This unique arrangement gives silk its exceptional combination of flexibility and tensile strength, making it strong enough to hold weight yet soft enough to be worn. Third is the efficient folding and compact structures. In many globular proteins such as enzymes and antibodies, beta sheets often form the highly packed hydrophobic core. This impact folding not only stabilizes the overall structure but also allows protein to perform highly specific biological functions such as catalysis or molecular recognition in limited cellular space. Lastly is the protein-protein interaction. Beta sheets also create broad, flat surfaces that are perfect for protein-protein interactions. These surfaces play essential roles in biological processes like immune signaling, DNA repair, and cellular communication. Without beta sheets, many of these interactions wouldn't be structurally possible. When these sheets form improperly or excessively, the results can be toxic, leading to serious sometimes fatal diseases. Misfolded beta sheets are the core of several major neurodegenerative and systemic disorders. First is the Alzheimer's disease. In this condition, fragments of a protein called beta-amyloid misfold and clump together into dense plaques. These plaques are rich in beta-pleated sheet structures and are highly toxic to neurons. Over time, they disrupt communication between nerve cells, leading to progressive memory loss and cognitive decline. Second is the prion disease. In this disorder, a normal prion protein, which typically contains alpha helices, transform into misfolded beta sheet-rich form. This misfolded protein is infectious. It causes other normal prions to misfold as well. The result is a cascade of degeneration that leaves brain tissue riddled with holes like a sponge. Lastly is the amyloidosis. Amyloidosis is a broader group of diseases characterized by the accumulation of insoluble protein fibrils in organs and tissue. These fibrils are largely composed of beta-pleated sheets. When they deposit in organs like the heart, kidneys, or liver, they interfere with function and can be life-threatening if not treated. Next is diagnostic and therapeutic application. First is the histological staining. To detect these harmful beta sheet aggregates, clinicians use special histological stain. One of the common is Congo red, which binds specifically to amyloid fibrils and shows in green birefringence under polarized light. Another stain is thioflavin. T fluoresces brightly when it binds to a beta sheet-rich structure, especially useful in Alzheimer's research. Second is drug development. From a therapeutic perspective, beta sheets are a hot target. Many experimental drug aims to prevent harmful beta sheet stacking or to dissolve existing aggregates. Some use small molecules that interrupt hydrogen bonding between strands, while others use monoclonal antibodies to tag and clear the misfolded proteins. Through the immune system, these strategies offer hope for diseases that so far have no cure. And lastly, biomaterials and engineering. Outside of medicine, beta-pleated sheets also inspire innovation in material science. The strength and flexibility of beta sheet-rich proteins like spider silk have led researchers to design bioengineered materials for use in surgical sutures, tissue scaffolds, and even wearable electronics. It's a remarkable example of how biology guides technology.
So before we end this video, we do have a provided supplementary notes, just a little or just a summarized infographic of our topic for this recorded lecture. We have also provided an assessment form for everyone to take in order to assess themselves of how much they have learned from this lecture video. It isn't really timed or doesn't have an actual deadline, but it is very much essential for you to take it so that we can actually assess, and not just for us, but for you as well. So that technically concludes our lecture video. Thank you so much for taking the time to listen to our video, and hopefully you've learned a lot from it.
This has been The Folded Truth, brought to you by Group 9.
Stay structured, stay strong.
Stay bonded.
Did they just forget about me?